animals and plant cells most of the times. So for example, we know that viruses has this capability to send their genetic material inside the human cell, right?
Yes, sir.
Correct. Now, how do viruses do that? So this is a cell, and let's say this is a viral particle that sits on the cell. How does it sense its genetic material inside? Quickly. Sometimes it basically uses some viroporins. These are the channels like holes. Literally, some viruses make a hole inside the membrane and send their genetic material inside the cell. I told you about this when we were discussing AIDS, right, as a disease. So a retrovirus sends its RNA inside, and the RNA gets converted into DNA, and that DNA incorporates into the host cell. Correct?
Yes, sir.
Now, if I want to, sometime humans also want to do the same. For example, we also want to send a gene inside an animal or a plant cell, and we want that gene to express there. So plasmid vector might not work there because animal cells and plant cells do not have plasmid vectors anyways. Okay, but these disarmed pathogen vectors, for example, adenovirus. So this category of virus known as adenovirus, so this is what is used as a disarmed pathogen vector. So it already has the capability to send DNA inside the cell. We just remove that pathogen's genetic material, so it's disarmed. Now it does not have any genetic material of its own. Instead, what it has is the genetic material that we want to send inside the cell. So that's a disarmed pathogen. It will act as a vector, and because we have not killed the pathogen, we have just disarmed it. It will go and do its regular job of infecting a cell and sending the genetic material inside. Now, by infecting the cell, I exactly mean sending the genetic material inside. Once that genetic material goes inside the cell, then it starts translating into proteins, and those proteins are toxins or other proteins that make the virus again in a normal infections case. But here that DNA will not code for any toxic protein, but will code for the gene of our interest. So we will get benefited and not harmed by that kind of a viral disarmed pathogen induced delivery of DNA. So that's called a disarmed pathogen vector.
So, sir, it will multiply the same like it does in body. So it will get multiplied, right?
It cannot multiply. How does viruses multiply? So viruses send their genetic material.
No,
This
sir,
genetic material won't multiply. Oh, sorry, you're saying about the replication of
yes,
this DNA.
sir.
Yes,
Sir.
of course, and we want that to happen. No, we have sent our gene of interest. Let's say this is a gene that is that was missing. Let's say here. So we will use a vector, which is a viral vector, and we do it in case of gene therapy. I told you about gene therapy.
Yes, sir.
Yeah. So in case of gene therapy, what we do, we fix the missing gene. So sometimes how we do, humans are born with some rare genetic disorders where one or the other gene is missing by birth, and when you are born, after that gene is important for certain activities that cannot happen. For example, your immune system's development and regulation. So if that does not happen, that you are more prone to infections and death, and people die very early. So in that case, we use these viral disarmed pathogen vectors to send our gene of interest inside the lymphocyte cells, which then can be transplanted back inside the patient. So there we are not relying on plasmids, but we are relying on viruses empty viruses, kind of, where they cannot do any harm, but they will just deliver what we want them to deliver. Is that okay? Is it clear what is disarmed pathogen
Yes, sir.
vector?
Yes, sir.
Yeah. So adenovirus vector is one example. We also use agrobacterium, right? The bacteria for plants. These are pathogen vectors. Here we in agrobacterium we still use the plasmid which is present in the bacteria itself to put our gene in, but in adenovirus, because it's a virus, there is no plasmid inside. We just pack our gene of interest within the viral protein coat. Okay, so that's there. Okay, what's next? Other doubts?
Yeah, sir, I had
Sahel,
a doubt.
yeah,
Yeah.
yes, Sahel. Arpit, you do you have any other doubt?
No, sir, it's clear.
Okay, great. Yes, Sahel.
So there was a question MCQ on ELISA, right?
Yes, correct.
Yeah. So ELISA is like detection for pathogens, like PCR for detecting corona or AIDS or HIV. So
Just
ELISA
one
here.
second. Let me share the test only. If I have, yes, I have the test. I can share this.
The 7th MCQ.
Let me stop sharing this and share this screen. Yeah, can you see the screen, everyone?
Yes, sir.
Yeah. Okay, so in the first 4 questions there was no problem, right? No one had a problem. If anyone had, please ask. I'm going directly to the 7th MCQ that Sahel is asking about. Yes, ELISA is.
Yeah.
Yes, Sahel,
So
what was the
my
problem?
understanding, it's an example of detection. It helps in detecting pathogens, right?
Correct.
Yeah.
It
So,
helps in detecting pathogens. Yeah.
yeah. So these were options. I didn't get to I didn't I get the answer for it. So
Yeah. So
I
what
just
is
went.
so when I was talking about ELISA, I told you that's the antibody antigen interaction based assay. Simple. It stands for what does ELISA stands for?
Enzyme-linked immunosorbent assay.
Exactly, enzyme linked immunosorbent assay. So what enzyme linked means, it uses immune enzymes. So it's basically an antibody which is produced by immune system plus an antigen which is produced by the pathogen. If the pathogen is present in our body, we can detect that pathogen by creating an antibody against the antigens produced by that pathogen. And if they both link together and we get the interaction, which means that you are infected by that pathogen. So the answer would be A, using radio labeled second antibody. It's not always important to just use radio labeled. You can also use fluorescent or other kind of labeled. Radio labeling is a very old technique, and it's still used for it's very handy, but because it includes radio, what do you say, um, it has a component of radioactivity, so there are better methods now. But basically you need to label the antibody so that you can see the antibody interacting with any antigen. So it's simple. So do you understand, Sahel?
Yes, I didn't know about the radio label or labeling part and such. So I
Yeah,
got
so
confused.
even if I was thinking of removing this radio labeled part from here, but this is exactly taken from the NEET. So they used this word, so I just thought that now because you have learned it, sometimes what they can do, they can also write using fluorescent labeled antibody. That's also true. Using labeled antibodies. That's also true. Using secondary antibody, using immune antibodies, whatever they write. You can also, the option could also be where there is no antibody, but it's like ELISA includes detecting the antigens produced by the pathogens. Okay, so instead of antibody, it could be just the antigen. Okay, so there can be many options. So even if you forget the radio labeled part, it just says using second antibody. Yes, because it has nothing to do with RBCs. It does not use RBCs. Using complement mediated cell lysis, we are not, no, it's not complement mediated cell lysis. Addition of substrate that is converted into colored end product, that's not there. That also does not happen. So none of these three are there in the NCERT. So one clue is what you have not at all studied in NCERT will not be the right question. So in NEET, 95 to 98% questions comes just from NCERT, but there are questions that come which are not directly given in NCERT, but ELISA is given in NCERT, right? So whatever they have talked about in NCERT about this technique, these three were not there. You will not study, but antibody term you will study, right? It's antibody antigen interaction. So that also. So do read your NCERT's textbooks thoroughly. Is that clear, everyone?
Yes, sir.
So they will not give you 2 or 3 options which all include antibody to confuse you. Most of them. Like that kind of a question will be a very, very tough question and a very confusing one. But just looking at the previous year's papers, the whole point is they want you to know a lot of things, but you don't have to go into very utmost details of everything. That's what NEET entrance is all about. Any other doubt, Sahel?
No, sir.
No. Okay, great. Anyone else has any doubt?
So what is the answer for the 7th question?
7th question, the subjective, right?
Yes, sir.
Yes. So this is I think what is the advantages of molecular diagnostic over conventional methods? So it's simple. So molecular diagnostics, I think this is not from chapter 11, right? But I'm not sure where it is from. But see, molecular diagnostics and so conventional diagnostics are your blood test, urine test, stool test. So these kind of tests were called. Sorry?
Chapter 12, right? Applications.
Chapter 12, applications, right? So this is a theoretical question where what is the advantage of molecular diagnostics? So molecular diagnostic is this ELISA, PCR techniques, all your high profile throughput techniques that have now come as a diagnostic using X-ray, CT scan. So these are molecular diagnostic techniques. Conventional diagnostic techniques were blood test, urine test, stool test, etc. So the advantage of molecular diagnostic technique is that it can allow you to diagnose a disease very early on, sometimes even before the disease onset begins. So for example, this is a very, very hot field in research, that can we use some biomarkers in the body, because it's very obvious, it's logical, because biologically any disease will not just occur in a flash of a second, that today someone was fine and tomorrow that person got cancer. So that won't happen, right? Cancer takes very slow process to develop inside the body, and then tumorigenesis happens, and when it begins you will not even feel that there is something wrong in the body. For example, one of the most problematic cancers in females is ovarian cancer. So ovaries are anyway very prone to cysts and tumors. So around 70% of females have either a small or a big or something insignificant or multiple cysts in the ovary, because it's a follicular gland. It has this habit of making follicles, multiple cells clubbed together, so it can very easily also form tumors. So you must have heard of PCOD, polycystic ovarian disorder, right? PCOD, anyone has
Yes.
heard of PCOD? Yeah, PCOD is very common, but PCOD is not fatal, it will not kill you. But if ovarian cancer starts, the point is female does not get to know about it. By the time you will start feeling abdominal pain or you will go to a doctor, it has taken over much of the ovary and also surrounding tissues. So 70 to 80% of the damage has happened. So the problem is it's not like we cannot treat diseases, we cannot diagnose diseases also at very early stage. So molecular diagnostics are more specific and give us a hand on benefit to diagnose the disease very early on, where it can be treated or cured completely. So conventional methods are not very good for that. Okay, so that this was just the answer.
So that's only like 1 point, right? Early detection.
Yeah, so you can just 1 point is they are better, like they give better resolution in terms of diagnosing the disease. Second, they help you in very early detection of the disease, and you can just third point, right? Okay, this helps in
All
curing
right,
many
sir.
diseases.
So what I did was
boards is,
I wrote 2 examples. So I wrote like PCR and ELISA, and then
yeah.
I wrote
So
like PCR is used to detect HIV, and ELISA is used to detect
mm,
like antigens and all.
yeah, you're right. So you just give examples, but did you also write advantage? So the advantage is,
Yes, sir. I wrote about the early detection, and then I straight away
yeah,
wrote the
great.
example.
Yeah, that's okay. You'll get marks.
All right.
So see, also sometimes, so these questions which I have categorized, some of these questions I did not wanted to disclose it to you, but I sometimes try to be very, not very, but little cruel in in these tests. So the questions I give you in 2 marks, sometimes in the real board papers, if you go Delhi's board, or so state boards also, I take CBSE state boards versus other. So every state has different papers. So sometimes these questions are in 3 marks, and the ones that I give in 3 marks, sometimes they're in 4 marks or 5 marks. But I just want you to understand, you know, how will you interpret that same question, and what will you write? So even if you write that 1 point in a 2 mark, you will get 2 marks. But if you see the same question in a 3 pointer, so because I think Matthew's point was that it is just a single point answer. Will I get 2 marks for it, right, Matthew?
Yeah, exactly.
But the question is, will you get 3 marks for it if it comes in the real board, in your board's exam?
No, I don't think so.
So, but you have to, right? Because that is the question. What will you write? That is the answer, and the question is clear, the answer is clear. So that's what I'm saying, that as a student, you also have to realize there are formats of examination. This was a mixed format. So your objective questions are like T20 format in cricket. You have to be fast, solve it, figure it out if it is the right question, a right answer, and then tick it. So you have 1 minute per question. So I think there are 180, or how many in entrances in NEET, somewhere around 120, 150 to 180 questions, and you get 3 hours, which means 1 minute per question. So it's like a T20 format. But when you go to board, sometimes it's like a test match. You have to write, so for this question, for example, you have to write that molecular diagnostic techniques include XYZ, whereas conventional methods are this. So molecular diagnostic techniques have advantage over conventional methods because of the following reasons. One, they are more specific techniques to look for various organs. Just elaborate. You understand what I'm trying to say? Just be smart enough to
Yes, sir.
answer. You get marks in boards for writing elaborative answers, which are properly presented as bullet points. So and I can actually assure you that it happens, because the same answer, word to word, same answer, without any change, if you just write it as a paragraph without any space, breaks, bullets, etc., and very small handwriting, where and you write the same answer in an elaborative manner without changing any word, but write just bullet points before starting every sentence, you will get, the one who's writing it, presenting it neatly, will get more marks. That's how boards and school exams work. They also take into consideration not just the quality of your answer, but also how easy you have made it for the examiner to check it. So that's true. Okay, so keep your presentations neat. Just bullets, point wise writing the answer is always appreciated. Making diagrams wherever necessary, so that you don't have to write 3 lengthy paragraphs. You can make one diagram, mark certain things, and start writing in bullets that as shown in the above diagram, this. That helps you to get more marks. Okay, keep this in mind. And good handwriting, of course. I think I myself use this. That's why I'm saying that it works, because I think I got around 5 to 8 marks extra just because of my presentation. Yeah. Okay.
Okay, sir.
Anyone has any other doubts from
No,
the
sir.
paper or in general? Any other doubts in general? If not, then let me go back to sharing the whiteboard. Oh, what's happening? Why is it not sharing? Okay, it's shared. Can you all see the whiteboard? Yes, no?
Oh,
Yes, can you all see the whiteboard, everyone?
sir.
Yes.
So can you share that notebook, sir? So I want to tally something from the notes.
You mean the OneNote notebook?
The
Actually,
other
I
notebook
can.
in which you make that
Yeah,
schedule.
actually, I'm not able to access it today, so that's why I'm teaching on whiteboard. But is it okay if I do it in the next class? Is there? You can ask me if there is anything specific.
No, no,
I can
no.
answer it.
I just wanted to check the order.
Order of what? Tell me. I'll tell you the order. Order of?
So like, sir, I didn't make the notes while the class, so I was making from the lecture. So I didn't know. Like I did.
Okay. Okay. So if you can send that, so you can do one thing. You can send the topic on the WhatsApp group, and I can send you the screenshots or the PDF of that portion of the notes.
Okay, sir.
Okay,
Yeah.
that will work, right?
Yes.
Okay, great. So just 1 second. So today we have to finish PCR first and also downstreaming processing to obtain the gene product. Now first let's begin with PCR. But before we go to PCR, I would like to let's go through what we have studied till now as a revision. So it all starts with our gene of interest, correct? Quickly.
Yes, sir.
Yeah, and what do we want to do with this gene of interest? We want to isolate it. So to get it, we have to do two things. One, we have to do isolation of DNA. And this I think you must have done as your school practicals for biology. I think that this is in your syllabus, right?
Yes, sir, I did that.
You did that? Great. So would you like to tell us how did you isolated the DNA?
Yes, sir. So
And also I'll ask you some critical questions that you have to then defend that, because I doubt whether you isolated the DNA. Okay, always doubt if you're not sure, let's say. So let's start with her experiment. So what did you do to isolate the DNA? Others can
at
also contribute
first,
to help. Yes, first.
like we have to take a fruit cell.
Fruit.
Not fruit cell. Fruit cell. Take
How
a
did you
sort
take a
of
fruit cell? Just one cell?
No,
No, I think
no.
you
You
meshed the
have
whole
to
fruit.
Sir, we also did
Yeah,
this.
yeah. You have to
Yeah, which fruit?
match.
Sir,
I
we
took
used
banana.
papaya.
You have to take a
Papaya.
soft fruit. You can take any fruit.
Yes,
So
banana or papaya. I'm also writing. Okay, just give me 1 second. So
Okay.
banana or papaya, then you mesh it.
Smash the fruit
Meshed up fruit. Why are we meshing it up?
so that the cells get separated.
Cells get separated, and in the process cells also can get damaged and open up, right? We want to break the cell
Yes.
open. That's
Yeah,
also
so that
happens.
we can get the DNA out of it. So
Yes.
after
So
that,
after
there's
that,
a lysine solution. Lysine solution was already prepared. It had shampoo and NaCl.
We use dishwash.
dishwash,
Yeah,
any
I
detergent.
guess.
So
Yeah, I guess you can use anything like that. But
great.
then it was
So
called lysine solution in our lab.
yes, so the
It was
lysine
already.
solution is nothing. So basically, okay, so go slow, Fatima. Okay, you have to lead us. So I'll just stop you at every point and just explain certain things. Okay, so this lysing solution simply means you have to lyse the cell, burst open the cell so that all the components of the cell come out, including the nucleus. Now in labs, in research labs, there are, for this lysing solution, you use enzymes depending on what kind of cell you want to take the gene of interest from. So the simplest is you take any soft fruit or any soft animal tissue and just mesh it and use detergent. So detergent breaks open the plasma membrane and everything comes out. But you cannot use detergent for everything. Sometimes it will also tamper with your DNA that you are extracting. So what you use is different specific enzymes like lysozyme if you are working with bacteria. So you would also like to remember this. So keep writing. If you are working with bacteria, you will use lysozyme. It's an enzyme that lyses the bacterial cells. Okay, so when we work with bacterial cells and we want to take out any bacterial gene, we use lysozyme. You use cellulase if you're working with plant cells, if you want to extract your DNA from this. Cellulase because the cell wall is made up of cellulose in plants. It's a cellulosic cell wall. So if you break the cellulose, the cell will open. Okay. And if you are working with yeast or fungi, then you have to use chitinase because
So,
the fungal, yes.
so we didn't use any lysing solution. So our teacher like took salt and then mix it with the soap.
Mix it with the
Something like soap solution.
exactly. So soap is the lysing solution. Okay, so
Soap
these are
with
all,
salt.
yes, soap, salt, detergent, they are all lysing solutions. We add salt to just enhance the functioning of that detergent and make it more polar, like make make the water ionizing, so it lyses the cell pretty fast. And salt was specifically added to precipitate the protein because we don't want protein contamination, right? Some schools don't use salt, some schools use salt. Salt should be used all the time because salt precipitates protein, but again, not all the protein. But if you increase the salt concentration, proteins do precipitate, so they are out and you get better purified DNA. So this lysing solution, these are the specific ones, and they are very expensive. So schools would not like to spend that amount of money because schools run on your fees only. So, and to do this one experiment, you can very simply use a very desi jugaad technique. You use any detergent or soap, hand wash, liquid
And
detergent.
sir, the best thing over here is that we are not going to see the genes or anything. This is just isolation of
Yeah,
DNA. So even
this,
if
yes,
the
yes,
genes, anything happened to it, it doesn't matter to us.
it doesn't matter. But you should know that in a real biotechnological experiment, it is the most crucial thing. So you do not worry about money, you worry about the purity and the quality of your DNA which you have isolated. So here detergent is the lysing solution, first thing. You understood, Arpit? Your teacher also used soap, which is a lysing solution. Okay.
Yes.
Okay, great. Then what you did, Fatima?
So this was added to the mashed banana, and it was
Yes, and
mixed
then you,
properly with the banana so that each and every cell gets
yeah,
broken. And
yes. So
after that,
what I did was my teacher, if I remember it correctly, she put everything. This meshed banana. We also use banana, by the way. We use meshed banana, put it in a airlock, ziplock, plastic
yes, I
ziplock,
did
Oh
the
yeah,
same.
we did the same. Yeah.
and
So we
then
did the same thing.
she circulated that to everyone to just beat it. You have to just beat it, but you have to be harsh enough so that everything get meshed up and mixed with the detergent, but not so harsh that the bag burst and everything comes out. So you
And
have to
not
beat
so
it, and
harsh
yeah,
that the components inside also, die.
components inside means
Means like the
inside the cell.
yeah, inside the cell. They can
Inside the
also
cell
get
nothing is
damaged.
living, so
No, they can get damaged.
there's only one thing we are concerned about, that's DNA, correct? And anything, whatever carbohydrate, protein, whatever happens to them, we don't care. So you can be violent enough. And about DNA, as you said, we are not interested in taking every DNA thread intact, even if it breaks into 2 parts or 3 parts, does not matter, right? At the end you will get it, right? So the whole experiment is about DNA isolation. Okay, so in schools, the fundamental, you should understand the fundamental. The way it is done, the harsh or soft, that can depend on why are you doing that experiment. Yes, so let's say it's done. Then what you did next?
So then we have to filter it
I'm
through a filter
saying
paper.
Yeah, I will also write that added salt. And this you should understand, this was to precipitate proteins. Okay, because we don't want protein contamination. Okay, now here, to get rid of protein contamination, salt is again very easy thing you can use, very easily available, accessible things in home or in your labs. But in biotechnological experiments, we are very serious. We do not want any kind of contamination. It should just be the DNA. So there are again different kind of enzymes used. Okay, so you can write down to remove contamination. So if it is a protein contamination, so salt precipitates protein. So protein becomes bulkier and goes down, does not comes up with the DNA. That is, but protein is still there, right? And there are DNA binding proteins as well. You know that. What will happen to them? They are already in a bound state with DNA. So do you think by that just by adding salt you will get rid of those proteins which are already bound to DNA?
no.
No, right? So, but we would like to get rid of those as well. So we use an enzyme called protease, right? Peptidases, proteases. You have studied about this in class 11th in your digestive system chapter.
We also studied it this year.
This year,
Protease.
okay. Proteases, correct. So, and for lipids, what will you use? Lipases. ASE is the enzyme that degrades it. So lipases. And for sugar, carbohydrate, you can use amylase, right? Salivary amylase breaks down glucose in our in the mouth, and then you also have studied about pancreatic amylase, peptic amylase, right? Yes or no? And when it is done in laboratory, we are so specific that we don't just want to get rid of protein, lipids, and sugars. We also want to get rid of RNA. So we
You can
just
use
need
RNAs.
Yes. So we use RNases. Okay, and these are some RNases are very, very important, because if you're just if you want to isolate DNA, the whole technique is to isolate nucleic acid, and RNA is also a nucleic acid, so you want to get rid of that as well. But in your school experiment, you just added salt. You did not specifically get rid of any of this lipid, sugar, RNA, etc. But still it will work. And why it will work? Because what is the next step? Okay, so here I have to add a page, so I have to go to the next page. So what is the next step?
Filtering.
Filtering.
The
Yes,
solution, you have to filter it through cheese paper, and
yes.
in the beaker you will get a solution without any pulp.
So what to keep and what to remove? Keep and
You can
discard
throw
after.
the pulpy part and you have to keep the solution.
Discard the pulp, right? Pulpy substance, which is the fruit pulp, and we have to keep the aliquot. We call it aliquot or solution. Why we keep the solution? Because we now believe that by now we have broken down things, and the components of cell is in this solution, including our DNA. Okay, everything else is just the structural thing, like the cell membrane, cell wall, the bulk of the cell, etc., etc. We want to get rid of it. So we get rid of it. Great. Then next.
So then
When
we
you
have
put
to
this
take
What we do?
chilled ethanol in a
test tube.
petri dish.
Yes. So you have to actually take the solution, okay, and pour chilled ethanol in it, right?
Yes, sir.
And you must have noticed that this step is often done by the teacher.
No, sir, we did it and we
We
had to
did it
like do it slowly through the
Yeah. Oh,
sides. We're
great.
not supposed to
that side
directly
of the
pull.
test tube.
So we
Oh, I
did
think
ourselves.
oh, great, great. So I think that's the best thing. So, and why chilled? Why this word chilled? What if it is at the normal room temperature? Why chilled? So what you have to do is you have to take this test tube. If you're taking this test tube and your solution is in the test tube, let's say, so you have to pour very slowly from the edges so that it goes touching the walls and makes a layer here, right?
Yeah,
You should not mix the tube or agitate the tube. If you agitate the tube and mix the solution, the ethanol with the solution, it's gone. Your experiment is gone. You have to do it again. Okay. So the whole thing relies here. This is called
So what
ethanol based.
happens if
Mhm.
it gets mixed?
If it gets mixed, then because ethanol is miscible in water, okay, but our point is we want to make a layer based on densities between the solution that contains everything from a cell and plus water and the ethanol, which is a spirit. So it is less dense and it is lighter than water, okay, but it will get mixed with water. So the point of pouring very slowly is to just allow it to settle on the top of that solution. And what happens is, let me draw it. So this is a bigger version of the test tube. So this is your solution where you have all your substances, your DNA threads is also present in it. And let's, when we are pouring, just 1 second. Let me use another color, different color, let's say greenish. So when we slowly pour ethanol from the sides, it goes and starts forming a layer, right? There will be a zone where ethanol will try to mix with water.
yes,
But because ethanol itself is you are using a chilled ethanol, anything that is chilled, its miscibility decreases. Like the, you know, you must be knowing that if you cool down substances, they cannot mix well, correct? So this chilled ethanol only helps the DNA particle. So all those DNA particles, I'll use red as DNA particle, which are present here in this solution, will start coming up and getting dissolved in this. Because DNA is soluble in ethanol, you will get your DNA particles here, your DNA threads here, versus protein, lipids, other things will just stay inside in this part. And if it get mixed, what Fatima was asking, if you do an experiment where you were pouring and you mixed it, so everything got mixed, so your DNA will just remain mixed in the solution. You understand? If ethanol is mixed with the solution, your DNA also remains mixed with the solution.
But after that, if we add more ethanol, then will it appear again?
After that, if you add more chilled ethanol
Chilled ethanol.
slowly, yeah, slowly again from the corners, then the problem is there is already ethanol mixed in the solution. So next time when you will pour, it will not be very effective in forming a layer, because it was forming layer on the basis of, because ethanol is a spirit lighter than water, so not lighter, actually heavier, but it was a spirit which was more volatile, so it sits on the top, okay. But if you already have ethanol here as well mixed, so it is now not water, not a solution of just containing water, it is a solution of ethanol and water, and ethanol will keep mixing, correct? If how slow you pour, this is what I have observed. So you do it you do it correct the first time, or most of the time you have to repeat. Okay.
Okay, sir.
And even if you are successful in forming a layer next time by using a very chilled ethanol, but remember ethanol is already here, that will not come up, right, and form a layer. Now it is mixed with water now, correct? It's miscible. So it will also keep DNA mixed with itself. So you will get very few in your layer. You understand? As compared to this, where you have a clear layer of
And so
ethanol and
how will that very few come? Matlab, what?
some
How will
will
it?
still, because some they will be at the very surface, no, at the surface where you have managed to form another layer which is not as good as this one. So some will come and get mixed here, but the yield will be very little, because ethanol is already mixed in the solution, and there will be DNA mixed in ethanol and will remain everywhere. Whereas in this case, ethanol is just in this layer, so DNA has more and more DNA can come in and be mixed. Make sense?
Yes, sir.
Okay, so this is ethanol layer, and then your DNA, as you said, your DNA gets mixed. Sorry, DNA is soluble. Oh, I used another one.
So why is it that mostly it comes in the form of bubbles, and also some people don't get the double helical structure, only just bubbles?
How do you bubble? So you have to not allow bubbles to form, because formation of bubbles makes the whole process very less yielding. So you should not form bubbles. That's the whole point. You should try to get rid of bubbles as much as you can. So if your tube contains bubbles, then it's a problem. Then your ethanol will not be very effectively solubilizing the DNA, because DNA might just be on the surface of the bubble or inside the bubble, or it will
So I viewed my DNA. Okay, it was very beautiful. Firstly,
you cannot
it
see your DNA actually. That's where I
no.
believe that schools always teach you the wrong thing,
Mm.
sugar coated as the right. It's not possible to see DNA. There's only one way to see DNA, that can be done through electron microscope, or through a very super resolution microscope. You can see DNA threads. So that's my question. The white
So,
thread that you get at the end of this experiment, what are those?
so what is that? How?
Exactly.
What
So
is
when
happening
you
here?
sorry to burst you. That's DNA, of course. It contains DNA. It's not like there's no DNA in it. But sometimes when teachers explain, they say that, so let's go to the final step. Let's go step by step. That's why I think it's very important for you to know and understand. So you take
So
the
I
solution
even
over
got the double helical structure.
what?
Yeah, I'm not kidding.
How did you verify the double helical structure?
I even showed it to my teacher, and I could see the double helical structure.
You cannot see your DNA, first of all.
I could see the double helical structure. I don't know if it was
Yeah,
DNA or whatnot, but then
it
it
was
was
just
bubbly
two
bubbly, and it
threads that
was
you get entangled like this. It was not be true, it will be multiple. So what you saw is more than one, and you think it's double helix. So first thing, DNA cannot be seen with the naked eye. Second thing, you will never get to know with just the spooling. When you separate through spooling, you put something inside and you try to take those thread like particles out of the solution, correct?
yeah.
That's called spooling. Through spooling,
So
you will never get to
what
know
if
whether
you
it's
observe these threads under the microscope?
you will just see threads. You will see a mucus like threads. That's all. If you see it under a super resolution microscope, so you will see that there are lots and lots of lipids and proteins associated and tangled with it. So it's full of impurities. And if you go under electron microscope, then you will see that you have a DNA structure. If DNA could have been, remember, if it was so easy to see DNA double helix structure, why will you give a Nobel Prize to Watson and Crick to figure out that DNA is double helical?
Exactly.
If it
Okay,
was so
so
easy.
sir, but then
Yeah,
can you please tell me then how did I see
exactly.
the
I will
double helical
exactly tell you
structure?
that. So let's go to the next point. Next step. So what you did, you put the chilled ethanol, then, because you are
Then I
leading us, right?
waited. Yeah.
Then you
Then
waited.
I
So
waited.
some weighting. That weighting was to just allow all the DNA which is present in the solution to get inside the ethanol. Correct. Then
And
what
then
you
the
did?
observation was, that's it. And then the observation was most of the people got like milky white precipitate, which was bubble type. But
Mhm.
I got
Threads.
not threads, but it was like bubble in the form of double helix.
Okay, if there is bubble, first of all, your experiment is not clean. Okay, sorry to say that, but that's true. So you first have to avoid any bubble. So Matthew, what happened to yours? Were there bubbles in your experiment?
Not really. It is more like a fiber-like structure.
Fiber, right? So if you get threads, that's a very clean experiment. Okay, these spooling. So when we spool, what you do is you put something like this.
So we used a
inside.
glass rod.
Glass rod
Yeah,
or
to
anything.
spool
You can
it.
use the tip of paper clip, which is bent. Okay, that also helps. So something which has a bent helps more. So you see that there are threads like things floating. You try to just touch a few, and then you slowly just twist whatever you are holding, right? That's called spooling. So you're trying to get all those threads out of they are mucus thread, like very mucus like threads out of the solution. Correct? That's called spooling. And it contains the DNA. But
So basically what I did was, after I spooled it, what I did, I mixed it again and I still got more fiber.
you mixed where?
The
ethanol.
solution, the thing. I didn't add
You
anything. I
mixed
just
the
mixed it again.
solution. Yeah,
Yeah, I just shook
then you
it
must have got
and I
more
got it again.
contamination of protein and
This is so
lipids.
funny. This is so funny. How can you get
Yeah,
I
after
know,
you
I
can,
got
you
it.
can. Yeah, you can get it, because all what I'm saying is, in our school experiments, when we spool DNA, we are not just spooling DNA. It is a lot of contamination with DNA, protein and lipid contamination. So when Matthew again mixed the solution, he again allowed a lot of contamination to come and become and precipitate in the ethanol component. So if you add more ethanol, you can mix it, and here I have used very less ethanol as compared to the solution. In your labs, you can use more ethanol as compared to the solution, right? If you use more ethanol and you mix it, still ethanol will allow some precipitation to happen. You will get threads. So you will think that, oh, I'm getting more and more DNA out. But over time, you are getting less and less DNA and more contamination out. Okay, so
Can we
that's
see the
That
something,
makes more sense.
because and that's what school teachers don't tell, because they just want you to feel that you have extracted DNA, which you have actually you have done it. So if you send it for any nucleotide test, or you check for nucleotide ratio in this, it will be high, very high, because you successfully taken out DNA from the cell had a lot of things: cellulose, membrane, plasma membrane, endoplasmic reticulum surface, Golgi bodies, nucleus, lysosomes, a lot of things full of tons of proteins, tons of lipids, but you very successfully could get DNA, which is majorly DNA. Even if it's 70% DNA, 30% contamination, you have done it. You have just got the DNA. And that's all what the purpose of this experiment was. But one thing that often students take wrong from this experiment is, when they see these threads, they think that they are DNA that they are seeing, right? These are DNA threads. They are millions and millions and millions of DNA fragments stuck together and precipitated like a mucus like long thread. So millions and millions and millions of DNA fragments are stuck together, and also there is some protein some protein which is already bound to DNA, some lipid content might also be there. So when you see that, if you see something just like this one, and if you just zoom it out, it will not be something, it will just be like millions and millions and millions and millions of things. If you zoom out even more, you will see that it has protein contains a lot of things. So what you are seeing as one thread is not DNA. It's not that easy to see DNA. We cannot even see a cell with the naked eye. To see a cell, you have to go to a microscope. Then inside the cell, there is a nucleus. Inside that nucleus, there is chromosomes. Those chromosomes are protein plus DNA bound on it. Then you remove all the protein,
So,
you see one DNA. Yes.
Actually,
tell me.
even I thought about this, like how can we see the DNA? But then
But you
I
did not
thought
ask
that,
your. Yeah.
but then I thought it got amplified somehow. I don't know.
You were not doing PCR, right? We will do it, Abhi. You did not amplify anything. You just got whatever was present naturally in that sample. Okay, and there was lots of DNA in that sample because you meshed one whole banana. That one banana might be having
Not
billions
one
and
whole banana
even if one
slice.
more slice, that one slice will also be having millions and millions of cells, and every cell will be having around what is the chromosome number of banana, 14 or something. So every nucleus will be having around 14. A banana is diploid, so 14 into 2, let's say 28 threads. So 28 into whatever million of cells that slice contains, that many DNA threads were there present in it. So you tried to, what you did is successfully you allowed it to precipitate out using chilled ethanol, and then as a thread you spooled out all those stuck together DNA threads. Okay.
So then how do we take out the DNA actually then? Also, sir, I
Yeah.
sent you a picture. Could you please look at that? That's how I got it.
Yeah. So where is that picture? Where have you sent me? Is it on
WhatsApp.
WhatsApp? Okay. That's great if you have captured.
No, I
Yeah,
drew
that's
it.
yeah, okay, you drew it. Okay, I think that's just an artifact that you know, sometimes we look in the clouds and we think there is a horse, but that cloud is not a horse, so
No,
it's
sir,
just an artifact that you must have seen.
I'm
Yeah,
telling you, I waited
I
for like
I agree that you see a
after I
double helical
completed, I
structure,
waited for my whole recess to be over, and then I
Mhm.
saw that.
But anything which is macroscopic and is two threads can just entangle into a double helix, no? For that matter, you know, my friend, one of my friend has curly hair, and whenever she keeps her hair open, some of these her hair strands go this. So is it a DNA? I have seen a double helix of her hair. So you saw a double helix of these precipitates. Simple. That's not a double helical DNA itself. You understand that? Because a double helical structure of DNA cannot be visualized by any microscope, except for if you go for electron microscopy. There also you will not be able to see the double helical structure. You will just see a thread bead, beads on string structure I told you about. That was an
Yes.
electron microscopy structure. So what did you see? You see some bead like things on a string like this, right? This is what you got to see from a electron microscope. Can you imagine? It is working at the resolution here is at the level of electron. So how do we get an image through electron microscope? Is all electron dense region will come as dark and electron light region will be less darker. So this thread itself is DNA, but no one knew that this DNA is actually double helical. This you cannot even see with an electron microscope, so forget about with the naked eye. Or
So what did
so
I
that's
see?
what you just saw. Two threads precipitated, they must have entangled in a double helical form. That's it. Just like I saw my friend's hair like this. Sometimes these two, if you have curly hair, these two hair will just go in opposite strands and make a double helical structure. Daenerys Targaryen had one meme on this. If that's one character from some.
So I feel so disappointed.
Science is very. That's why I think school teachers don't want you to get disappointed. They want you to feel that joy. Oh, we extracted DNA, which you did. You did extracted DNA, but that was not the DNA that you are thinking. Like it is millions and millions of thread, not purified with lots of contam. Is that okay, everyone?
So,
This is how. Yes, Arpit.
so what if we, so our teachers like blended the fruit, so like we will get the
Yeah.
D.
Yes, of course. So blending just helps. For example, if you're doing it with pineapple, like I think you said you use pineapple, right? So pineapple is harder than apple and papaya. If someone is using pear or pineapple, it becomes difficult. So even if you blend or put it
We
in the
will get
mixer
the
and
same results, right?
yeah, you will
It
get the
won't
same
affect.
result.
Oh,
Not
okay.
much, not much. Won't affect much. In fact, you might get more and more threads like Matthew get if you are blending the fruit. That can happen because after some time more and more content starts coming out. Okay.
Oh, yes.
I'm so sorry to disappoint you, Fatima, but I hope you did. You understand now, right? The whole point, the concept. See, someone got a Nobel Prize to figure out that DNA is a double helical structure. Scientists scratch their heads for decades to just figure out what could be the structure of DNA. What is actually DNA? And if it was this easier, then just imagine how dumb Watson and Crick would look now. That school students are just, precipitating it and saying that, see, this is a double helix, this is a single helix, and what you were doing. So it's not possible. Even at the level of molecule, if you think, it's not possible to see it. But threads, when millions of threads are together, they make a bigger thread. It's like when you see a rope, do you see the individual particles of that rope also? So rope is made by finer threads individually, right? Or when you see pleated hair, someone has make pleats of the hair, so that looks like one strand, right? One rope like strand. But do you see that? Do you see individual hair in that pleat?
No.
And that individual hair also has a structure. So what you saw was a pleat, and you think that and those two pleats were just bundled together like a double helix. That's all. It's an artifact. But in that single pleat, there are millions of finer particles which are hair like threads. That is your DNA, which you will not be able to see with naked eye or even
So why
with a
did we
microscope.
have to come into a double helical structure to make
It just
this
happened. That's what I say. It's an artifact. It just
It
happened.
was your bad luck.
No, it was a good luck probably. Yeah, maybe you just got tricked by, like I said, no, sometimes we look in the cloud and we see a horse. So you looked in your thread and you see a double helix. You can just get a double helix with hair strands or with any two thread strands for that matter. It's okay. Okay, great. And threads often, when entangled with each other, they're entangled in a manner like, see your earphones. Most of the times my earphones are double helix strangled, right? Something that strangles over itself and got entangles over itself will give you a look of double helix. No, it's not that difficult to just think. So don't be too harsh on yourself. You did a good experiment, but you're not just inferring it correct. And that was exactly my worry. And it happens every time I talk about this topic. Every time I ask what you did in your school, they often have this false perception that they saw a DNA thread. You saw a pleat, okay? You saw a rope.
Sir, one more thing that you said,
Yes.
that this thing that we saw, it contains many other things also. So how
Yes,
do you
because
remove those many other things?
by
see
using these
Mhm.
by using these enzymes, oh sorry, I have to go to the first page. By using these enzymes, proteases, lipases, amylases, and RNases, you have to treat your final solution with these so that every protein, lipid, sugar molecule gets degraded and broken down into pieces so that it cannot stay attached to the DNA or it cannot come out with DNA.
So one thing. So if we do this after we get the solution, and then afterwards we treat it with chilled ethanol, then what will happen?
Then you
Can
will
we
get,
see?
in the best case scenario, you will get pure DNA threads, but again those will be millions of DNA threads stuck together, and what you will spool out will be a thread like structure which will contain millions of DNA molecules. If you have no other contamination, it will be less mucus like, less slimier, and more whitish, not whitish actually. If it is white or if it has some color, it clearly means it has contamination, because only proteins and lipids have some color kind of thing in a solution. DNA has no color, right? So if you are getting it a very whitish thread, it clearly means it has contamination for sure. That's why you can see it. So remember, I told you that to visualize a DNA, we have to stain it by ethidium bromide.
Yes, sir.
if DNA was just visible like a white thread, why would we use Ethereum bromide then? We can just see the DNA, but we cannot see the DNA. It has no color. Okay. So in labs, when we purify DNA, we do not get anything. We do not get any color. We just, when we super, so instead of spooling it out through ethanol, we do something called super centrifugation. You will get a pellet, a small dot kind of thing that will look slightly whitish, because again it will have some contamination. But then we mix it, we treat it with these proteases, RNases, lipases, amylases, whatever you require, and then you check whether you have DNA or not on a very, very small, just as big as your, what can I say, a small suitcase. It's just as big a machine as a small suitcase, but it costs $1 million, lakhs of rupees, because it's called a nanopore. So in that nanopore, you put 1 nanoliter of your solution, 1 nanoliter. Okay, like just 1 drop, not exactly 1 nanoliter, sorry. You put 1 drop of your solution and it will tell you a concentration of DNA or nucleotides in that 1 drop. So it's called nano drop machine or nanopore machine. So that's how we get to know that whether we have done the experiment right and we have precipitated DNA only or not. And we don't use chilled ethanol. We don't only use chilled ethanol in labs. There's something called trizol, which is also used if you want to go. It's called trizol based DNA isolation.
So it
Or
is so
RNA
hard
isolation.
to get the gene of interest.
It is so hard, exactly. You know, during my thesis, my MSc thesis, I scratched my head for weeks to get the DNA to make a cDNA out of it. Not actually DNA. I was working with RNA, even harder. RNAs are molecules. You will isolate RNA, you will look the other way, you will look back, and your RNA is degraded. You have RNases everywhere. RNases are so robust enzyme. We have RNases on our hand, in our saliva, in the sweat, in the tear, in the air, everywhere, in the hair, everywhere. On a dust particle, you will have RNases everywhere in the nature. So to
So how
take
do
out
you
RNA,
isolate them?
yeah, we have to keep the surface clean, you know, wiped with ethanol. We have to wear gloves wiped with ethanol, constantly keep wiping it with ethanol in the process. Wear a mask. You should not breathe onto your sample, because if you breathe, you will send a lot of things, some particles, aerosols, bacteria, any virus that will surely degrade the RNA. So no saliva, no breathing in the sample. Of course, there should be no hair strand falling into it. So if someone has really bad problem of, you know, dandruff, isolating an RNA will be very, very difficult for that person. Yes, so I was isolating RNA for my thesis. So it's called trizol based RNA isolation method. So it's complicated, but the logic is simple. Just the doing of it is complicated. That's why we need biotechnology and a lot of development in this field. But the idea is that DNA can be precipitated out using ethanol or trizol, and then you can. What is the next thing that we do? So any doubts with isolation of genetic material? I don't think we'll be able to do PCR today because this itself was very, very discussive. But yes, it was worth of it. You should know it. Any doubts with DNA isolation or
No.
nucleotide isolation?
No,
No.
sir.
Now what next? Once we have isolated the DNA, what is the third step? Sorry, what is the second step? What do you want to do with that isolated DNA? You want to use,
Get the gene of interest.
yes, you want to use restriction enzymes that you have decided.
So
And this
after
is,
all of this, I think it's not even possible.
sorry,
After looking at all of these processes and stuff, it looks so much impossible.
It is not impossible. It's a routine thing. If you do you see people taking insulin?
Yeah.
If insulin is in front of your eyes, this all happened in the backdrop. So we should always be thankful to people who work for us day and night. Everyone, like every kind of service you get has a lot of background processing in biotechnology. So if you see insulin, anyone taking insulin shots, all of this must have happened. That's why the insulin is there, right? They extracted the insulin gene, they extracted the DNA from a human cell, extracted that gene, put that gene in the plasmid vector, put that vector in the bacteria, allowed the bacteria to make insulin protein, burst the bacterial cell, purified the insulin protein, made it into a formulation, put it into these injections, and then people take it and manage their diabetes. So it's very, very doable. We just did it in 7 months to make the fastest vaccine humanity has ever produced against COVID-19. It's possible. So next is you want to use restriction enzymes to cut your gene of interest from that DNA. Because remember, abhi you have just isolated the DNA. Then you cut your gene of interest. And then you use the same restriction enzyme to cut your plasmid to make sticky ends. Then you bind both of them. Okay, but there is a process in between which is known as amplification. Sometimes we would like to amplify our gene of interest. Amplification of gene of interest. Okay, and this amplification of gene of interest is done through a process called PCR.
PCR.
PCR stands for
Poly.
yes, polymerase chain reaction. Yes. Now there are three terms and simple. Polymerase is used because we use a DNA polymerase. Chain reaction is because it's cyclic. We are doing the same thing over and over again and multiplying that fragment of DNA of our interest. Now, in PCR, there are three components about which I would like you to go and read. So I'm giving you homework so that you are more clear and you don't have, see, you know, this is not to demoralize or to excite you. It's just neutral, a thing that in one go, it is totally okay to not understand science and the fundamentals of it, even if you understand what are we doing, because today's class is an evidence for that, and this topic in every batch is an evidence for that we do certain things but we don't understand what we are doing, correct? Or we understand it in a different way when we are doing it. We are understanding it completely different when we are doing it. We don't ask these questions, and if these questions are not answered, you will always be confused in your head that, oh, I could see, for example, I could see this molecule, but the question is, can you really see that molecule? Was it that easy? So what then? If you say no, then the next question is, what did you see then? That also has to be known, right? So the kind of questions Fatima asked today were brilliant questions because they teased out every fundamental. So she first said that she saw something, which when she realized it was not that, then she asked what was that, which I really appreciate. And she got to know that, okay, it was millions of threads, you know, bound with some impurities. Then her question was how to get rid of those impurities. Again, now these kind of questions should be asked to every teacher at every level who's teaching you. That's the job of a teacher. And then she asked that what if we get rid of all the impurities? Then you will get a thread which will not be very visible to you in that solution, right? That's the problem that we face. Then we label the DNA and see it. And that's the $1 million problem that Watson and Crick solved, and they got Nobel for it. Okay, about the structure. So today you have to go back and study about three things. One is DNA polymerase, the enzyme. Just the basics, brush it up. I have taught you about this. What does DNA polymerase do? Its direction and all, just revise that. Second is at what temperature DNA polymerase works. Okay. And third is about a bacteria which is called Thermus aquaticus. You can just go to any source, just try to take more reputed sources. Thermus